Hello, good afternoon, everyone. Okay, I hope you're all keeping yourselves well and hydrated. It's been very hot these last 2 days, yeah. So I hope that everyone is keeping well and make sure you drink a lot of water. So as you can see, it's very hot here, and I have my fan at full blast here. So you see my hair is flying all over the place. Okay, so today I'm going to continue with chapter 3. So today I'm doing 3.2, which is the concept of movement of substances across the plasma membrane. Okay, so I'm going to share screen now. Okay, I hope you can see this. Yeah, all right. So this lesson, I'm going to explain the types of transport, which is the active transport and the passive transport. All right, okay. So you can see this, I hope, yeah? I think so. Right, let me start by explaining, yeah? What are the characteristics that determine the molecule can pass through, okay? Through which method, what type of transport will allow the substance to pass through, okay? So let's look at this one. There are characteristics of substances that are able to move across the plasma membrane. So it is determined by 3 factors. Okay, what are the factors that determine whether a molecule can pass through or cannot pass through the plasma membrane? Okay, so number 1, the first factor here is the size of the molecule. So whether it's big or small. So you can see here that I have 2 types of molecule here. So the one on the left is glucose, okay, which is a rather big molecule. And the one on the right here is the water molecule. You can see the H2O. So the difference here is in terms of size, all right? So size will determine whether the substance can go through the plasma membrane or the phospholipid bilayer or using another method, all right? Okay, the second one here is, all right. The second factor here is polarity of the molecule. So whether it is polar or non-polar. Okay, now polar doesn't have the same meaning as charge. Okay, so if you see this polar molecule, the one on the left here, this is call it as hydrochloric acid, which is HCl. So you can see that towards nearer to the hydrogen atom here, or the other this side here, is more positively charged because the electron has moved over. The electrons are unevenly distributed, so they are nearer to the chloride atom there. Okay, so one side is more electronegative, the other side is more electropositive, but overall the entire molecule is neutral. Okay, so it's the same also for the H2O. It is a polar, it's uncharged but it's polar. And the other one, this is NH3, this is ammonia. This is a molecule of ammonia. Okay, for non-polar molecules, you have nitrogen gas, N2. CO2 is carbon dioxide. And this is tetramethane, okay? Tetramethane, yeah. CCl4, tetrachloromethane, yes, okay. So the next one here is charge. Okay, whether the molecule is charged or uncharged. So if you see here, there are 2 examples here. The one on top here is ammonia molecule, okay, which is uncharged because they share the covalent bond. They have the covalent bond between the electrons. So the electrons, the charge is neutral. Okay, the charge of the entire molecule is neutral. But for the ammonia ion, so NH4. So you have the extra of a hydrogen ion, all right? So you have this extra positive charge. So the one on top here, we call it a molecule, ammonia molecule, and then this one here is an ammonium ion. All right, so 3 factors: size of molecule, polarity of molecule, and also the charge of the molecule. So determines whether it can pass through the plasma membrane and through which method or through which part. Does it pass through the phospholipid bilayer, or does it need to go through the transport proteins? Okay, so let's see. Yeah. Now, what molecules can pass through the phospholipid bilayer? So you have to memorize this. Yeah. Questions will be asking what this molecule, all right, how does it pass through, or what molecules can pass through the phospholipid bilayer. They also ask you what are the molecules that can pass through the channel protein, and the other one is the carrier protein. So there are three, we call it pintu masuk, right? There are three ways, or what they call three ways or methods where you can pass through, all right? So let's look at what other molecules can pass through phospholipid bilayer. So these are the characteristics. Okay, as long as the substance has these characteristics, which I'm going to show you afterwards, they will be able to pass through the phospholipid bilayer. Okay, now what are the characteristics of the molecule? Okay, number one, the molecule has to be lipid soluble. That means they are uh, able to dissolve in lipid, or they are fat soluble. Okay, examples would be fats. These fat molecules are definitely fat soluble. Okay, when something is fat soluble or lipid soluble, it means it's not water soluble. So substances are either water soluble or fat soluble. So if it's not water soluble, it could be lipid soluble. And then if it's not lipid soluble, it should be water soluble. All right, okay, so all the molecules which are lipid soluble should be able to pass through the phospholipid bilayer. Okay, another characteristic, it is non-polar. That means it must not be charged. Okay, it is hydrophobic, right? It's not polar, and it's also not charged. Okay, non-polar and uncharged molecules. Okay, afterwards you'll see. Okay, now examples here are the substances which belong to this category: lipid soluble, non-polar, and uncharged. Examples: fatty acids, glycerol, vitamins A, D, E, and K. They are big, they are small. Steroid compounds, because their steroid compounds are lipid soluble. Okay, and another group of substances would be having these characteristics. They are small. Okay, they may not necessarily be lipid soluble, but they are small and they are uncharged molecules. Examples will be carbon dioxide, which is non-polar. Oxygen is also non-polar. Another one is water. Okay, so remember these three examples for small uncharged molecules that can pass through. Okay, remember this would count COW, carbon dioxide, oxygen, and water. Right? They are able to pass through the phospholipid bilayer. Okay, and the other group will be all the molecules that are lipid soluble. Does not matter whether they are big or small. Fatty acids, they are big. Okay, but because they are lipid soluble, they will be able to pass through the phospholipid bilayer. And then vitamins A, D, E, and K, they are small, but they were able to pass through the phospholipid bilayer because they are lipid soluble. So in conclusion, as long as they are lipid soluble, the molecules will be able to pass through the phospholipid bilayer. Okay, whether big or small is not a problem. All right, and then the other group will be small uncharged molecules. Remember COW, carbon dioxide, water, oxygen, and water. Okay, so you can copy this down later, yeah? Okay, when you review the lesson again, you can copy it down. Okay, next. Now, what about molecules that can pass through using channel protein? So those other molecules that do not qualify just now the characteristics, right? Lipid soluble, okay, uncharged, hydrophobic, and all that. They are not that group. Now, that means they must find another way to enter. They cannot enter through the phospholipid bilayer. That means they could have to enter using the channel protein or the carrier protein. Okay, let's see what are the molecules that can pass through the channel protein. Okay, so the channel protein here, now you must know how to recognize the channel protein. So the channel protein, you can see in this diagram here, is the one on the left. Okay, the one that is colored pink. Okay, the reason here is because channel protein will always have an open pore. The pore does not change in shape and so on. So the one on the left, you can see is always open. The pore is always open. So that is the channel protein. All right. Now, what are the substances that can pass through the channel protein? Just now cannot pass through the phospholipid bilayer. If they are water soluble and they are charged, okay, they will pass through the channel protein, or we call the pore protein. Examples: small ions, okay, potassium, sodium, calcium, magnesium ions, and all that. They are all charged. Okay. Another substance that can pass through the pore protein or channel protein is also water. That means just now we saw water, right? Water can pass through the phospholipid bilayer. water can also pass through here. So water can pass through more than one, more than one. It has more than one door. Okay, it can pass through more than one way lah, to enter the cell or to exit from the cell. Okay, so this is channel protein. All right. Now then you have another group of substances which do not qualify the first characteristic just now, and also does not qualify here. Okay, because they are large. All right. Now let's look at the next one. Yeah. So this one is channel protein. Now what about the third group? Okay, if it doesn't qualify the first, it cannot go through the first method, it cannot go through the second, which is the channel protein, it will be able to probably go through the carrier protein. Okay, so the third group of substances that will go through the carrier protein, okay, what are the characteristics first? First of all, they are water soluble. They are large water soluble. Okay, so examples of large water soluble molecules will be glucose, okay, the sugar, right? All these sugar molecules. And then you have amino acid and also urea. Okay, these are a few examples that you just need to remember. Of course, there'll be more, but you don't need to remember the entire spectrum. Okay, you just need to remember those which are commonly asked in your Form 5 or SPM. Okay, at this level will be those very common few examples. All right, okay, so you have already seen and also need to remember what are the molecules that can pass through the phospholipid bilayer, and then the channel protein, and then the carrier protein. Okay, you need to make a list and you need to memorize them. Yeah. Okay, so this is the carrier protein. Okay, now summary. All right, summary of just now. So I'm going to put a summary here. This is a picture from the textbook. You can look at it later. So the first one is, see all this put here, COW, okay, which is your carbon dioxide, oxygen, and water. They will be able to pass through the phospholipid bilayer. Okay, the reason is because they are not charged. Okay, they are uncharged and they are small, so they can squeeze through. They can squeeze through the phospholipid bilayer. Okay, then another thing that can pass through your phospholipid bilayer are those lipid soluble molecules. As long as they are lipid soluble, okay, doesn't matter whether they are big or small. Example would be fatty acid, glycerol, vitamin A, D, E, and K, and also steroid. They will pass through the phospholipid bilayer. All right, okay. Now, the second method where the substances can enter or exit the cell is through this channel protein. So ions, right? Ions means they are charged. Okay, it's all these ions like the potassium and the sodium ions and all that. So they will be able to pass through the channel protein, including water, right? Water are able to pass through here as well. Okay, then you have another group of substances which are large molecules. Okay, large molecules, they will not be able to go through the channel protein. They will, and also because it's water soluble, they will not be able to pass through the phospholipid bilayer. So they will have to go through another way, which is through the carrier protein. Right, examples will be glucose, amino acid, urea. Just remember these three, and they will pass through the carrier protein. All right, so this is the what they call the summary of just now on things that you need to memorize for your exam. Okay, let me carry on to the next part. Yeah, okay. So the next part here deals with the movement, the method of passing substances across the plasma membrane. So we classify them according to, okay, first of all, two types. One is a passive transport, the other one is active transport. Okay, so what it means by passive transport? Passive means that it does not require energy. It does not require energy for the cell to pull the substances inside or to pump it inside. Okay, it means it does not require ATP. And some of this can happen across not even living cell. For example, it can happen across a whisking tubing, which is a not living cell, right? You can enter through this membrane, which is not living, just like what they call the whisking tubing, okay? The cellophane. These are selectively permeable membranes. It doesn't have to be a living cell. So because it doesn't require energy. Non-living cells or non-living structures do not need energy or cannot produce energy. Okay, so this is passive transport. Now, active transport is the transport which requires energy. So this, in order for this to happen, it must be occurring in a living cell. That is the main difference here. Okay, active transport requires ATP, requires energy. Therefore, it cannot be carried out in we call this whisking tubing, because whisking tubing is a synthetic membrane. It is not a naturally occurring. It is not. It's not. It doesn't belong to part of a cell. Okay, it does not require, and it requires energy for active transport. So it will not be able. This whisking tubing will not be able to carry out active transport. Okay, under passive transport, there are three types of methods of transporting substances. Okay, the three methods are, okay, we're going to look at it one by one. First one is simple diffusion. All right, simple diffusion. The second one is osmosis. And the third one is facilitated diffusion. Okay, so you need to see the entire structure. So I want you to see that under passive transport, there are three methods. Under active transport, there's just one only, and it all requires energy. Okay, and this one also requires the carrier protein. Facilitated diffusion also requires carrier protein, but does not need energy. Active transport requires the carrier protein and also energy. Okay, now let's look at each one. We start with simple diffusion. Okay, I don't think there are any questions here. Okay, a few of you are watching, but I think there are no questions here so far. Let me carry on, yeah? All right. So first one, passive transport. All right. Now definition first. All right. Passive transport is the type of transport that does not involve the use of energy. This is the basic definition of why we call it a passive transport. Okay, it does not require energy. And then the analogy, that means it's something that is similar. You want to see the concept of passive transport. It's like riding a bicycle downhill, right? Something coming down by itself. If it goes downhill, you see this person, when you're riding a bicycle down, you don't even have to pedal. You don't have to put your energy to pedal the bicycle, because gravity is doing the job for you, right? You have a higher concentration. It's like substances that move along or down the concentration gradient. It's always from more to less, or what we call a higher concentration, okay, to a region of lower concentration. So that means one side of the substance, or the we call it water or whatever, one side has got more of this molecule, then the other part here will have less. So naturally, automatically, the molecules will try to move from the place that has got more molecules to less molecules on its own. You don't even have to push it. All right, it will do it by itself. So this is the analogy of a passive transport, whereby substances will move along, or we say down the concentration gradient. You can say move along the concentration gradient or down the concentration gradient. That means it's always high concentration to lower concentration. All right, okay, so this is just analogy to you to just imagine, okay, things will always move from more to less, just like higher ground to lower ground for your bicycle. Okay, examples of passive transport are those three. So all these three, simple diffusion, osmosis, and also facilitated diffusion, the molecules will be moving from a region of more to another place or another region that is less. Okay, more molecules, the area that has got more molecules will go down or move to the other place, the other part, which has got less molecules. Okay, so let's look at. Now we look at one by one. Simple diffusion. Definition first, simple diffusion. Simple diffusion is the movement of, okay, so it's a movement. All of these are referring to movement. So you must start your definition by saying it's a movement. Movement, number one, of what? What is moving? You're moving here, you can say particles, you can say molecules, you can say ions. Okay, it can be particle, it can be ion, it can be molecules. From where to where? That's important. Okay, it's a movement of particles or molecules or ions from a region, or we say area of higher concentration, means that place has got more of these molecules, to into an area or to a region of lower concentration, that means has got less of these molecules. All right, okay, so more to less. And for this simple diffusion, you don't need to have a membrane. All right, you don't need to have a semi-permeable membrane, or in fact any membrane. Okay, it's just a simple example. If I have, if I'm wearing perfume, let's say, all right, I walk into the class one day, or I spray myself with a lot of perfume, all right, and you find that when I'm standing in front of the classroom, the students in front, the front row will smell the perfume first, okay, because my perfume came from me, which I have a lot of the molecules of perfume. It was spread out to a region that is lower, which is your side, the student side, no perfume at all. So from my area, from my place here, which got a lot of molecules of perfume, it will try to move out and move to a place that has got no perfume at all, which is where the students are. All right, so this molecule, you can see the picture, the animation here. The green particles which were inside the bottle, when you open the bottle, these molecules will try to move out, we say diffuse, from a region of more, which is inside the bottle, to a region which is less, which is outside the bottle. Right? In the beginning, there's not even one molecule of the gas. All right, then afterwards you can see it spreads everywhere. All right, okay, so that is the definition. And these molecules will move down the concentration gradient, that means more to less. Now, when does it stop? Right? Now, it will move until both sides or both regions will have the same concentration. Okay, just now I said you start with one place which got more, the other place has got less, totally no molecule at all of this gas. So it will start moving until both regions will have the same concentration, and then we will say that it has achieved a dynamic equilibrium. Okay, dynamic equilibrium means both sides will have the same or equal concentration. Okay, when that happens, we say dynamic equilibrium is achieved. Okay, now the molecule will still be moving, yeah. Don't have the wrong impression, because they have energy. All these molecules have energy, so they'll still start moving. Okay, but because there's no difference in concentration, so maybe how many from this side move to that side, it will still be the same as the other side moving back to here. So the movement is still there, but there's no change in the concentration. Okay, both regions have the same concentration. Okay, so we call it dynamic equilibrium is achieved. Okay, so you can see here, when you have a glass of water in the beginning, there's no color, right? And you drop a color liquid on top, right? And then you see it start to spread downwards, because on top you have a lot of these yellow color particles, and at the bottom is barely any. You don't have at all. That means low concentration, and the molecules will start diffusing down. Okay, whatever, it could be iodine, it could be orange juice or whatever lah. All right, the molecules will start to spread. We call it diffuse. Okay, use the correct term. Don't say move, but use the word diffuse. All right, okay. Okay, hi, MUHG 49872, 4987Z. Okay, hi, hello, thanks for joining the class. Yeah, I hope you will learn this part. Yeah, I hope it's clear to you. If you have any question, you can just ask in the comment box. Yeah. Okay, so let's look at simple diffusion again. Now, simple diffusion is the method by which, this now I mentioned the substances that pass through the phospholipid bilayer. It passes through using this method. Okay, using this method, the carbon dioxide, oxygen, water, remember the COW, it diffuses across the plasma membrane. Across the phospholipid bilayer. And also just now the lipid soluble molecules like vitamin A, D, E, K, glycerol, okay, steroid, all these also will diffuse. Fatty acid, they also diffuse across the phospholipid bilayer through simple diffusion. You must mention the word. All right, okay. So it can occur with or without a plasma membrane. Okay, just now I mentioned, I said the perfume there. I was saying about the example of me wearing a lot of perfume or spraying myself perfume. Then I walk to the classroom, and then people in front of me can smell already. There's no even membrane between me and you, or between me and the student. So it does not need a plasma membrane. It does not even need a membrane. Okay, molecules will simply move from one place that is more to another place that is less. This is a universal law. When you have some place that's more, it will try to spread away. It will try to move away from where there's a lot, because it's too compact, right? It will try to move away to other areas which has got less. Okay, right? Wait, I think I missed out something. Wait. Structure A, where is it? What happened? Yeah. Wait. Why am I going back? Wait. Something happened to my PowerPoint. Let me see. Yeah, I'm going to share first. Yeah, something happened. Okay, never mind. I think I will just go through. This is all my lesson earlier on. I think I will need to go through. I don't know why it came back. I don't know. There's something wrong with the PowerPoint. 1 minute. I think I will just unshare. Because unshare. No lah. I think it will be too messy for me to unshare. Now this was just the previous lesson that I had last week. Right? Just to show you. Okay. I think I'll just skip through quickly. Just and all this you can just run through lah. Alright. If you have missed my lesson, remember you can always go to my channel to get the lesson. Okay, now let me go through this. Okay, this is today's one. Right, I think there was a glitch somewhere. Right, this was my lesson just now. Right, I don't know why it just went backwards. Okay, let me carry on with this. This is also done. Sorry, just a glitch here. Now we go to simple diffusion. Right here. So it can occur with or without plasma membrane. So examples of molecules that enter plasma membrane through simple diffusion. Okay, so it will be this. That's why I mentioned the one of the lipid soluble molecules. Fatty acid, glycerol, ADK, steroid, they will come in through simple diffusion. Another one is the oxygen and carbon dioxide. Sorry, not water. Water will go into another method. Another name. Water will go into another one. We have a different name for water diffusion. All right. So it's these substances: fatty acid, glycerol, ADK, and steroid, oxygen, and carbon dioxide. They will diffuse across the phospholipid bilayer through simple diffusion. Okay. Now next. Okay. Now next one, osmosis, the second method. Okay. Some tips. Okay, later maybe I do not know what your teacher. I think the best person to give you tips will be your teacher. Okay, because the teacher sets a paper. So I do not set a paper. Right? I will not know what kind of tips to give you. What tips I give you is all the things that you need to remember lah. The things that generally what is inside this syllabus, in this chapter, in this subtopic. Just like I told you what are the molecules that can pass through. Okay. As we go along, I will tell you what is important. That's all I can tell you because I don't set the paper, right? The best tip to get is always the teacher who teaches you, and he or she will be able to advise you what to pay attention to. Okay, so what I can tell you to pay attention to is through the requirement of the syllabus. This is important. This is important. I can tell you that. All right, but whether the teacher wants to produce the question on that or not is up to her, right? Okay, so let's look at osmosis. Now, osmosis refers to, the definition. Definition got 3 clauses, 3 short sentences, we call it. Okay, number 1, it is net movement of water molecules. So the first thing is what is moving. Osmosis is only referring to water molecules diffusing. You cannot use it, osmosis, to refer to ions moving or ions diffusing or other molecules, lipid or other substances, oxygen, carbon dioxide, cannot. You can only refer to osmosis. Only to refer to water. All right, okay. So for osmosis, I can tell you MUHG. All right, I can tell you what is important. So number 1 is a definition. It refers to what is the osmosis. Osmosis is the net movement of water molecules. Okay, only 1. Next one, another 2. Second sentence. From where to where? From an area of higher water potential. Higher water potential means got more water. So let's say you got 2 types of solution here. I've got A, I've got B, separated by a selectively permeable membrane, right? You see A has got more water on the A side, and B has got less water. You see, how do I know? Left-hand side got so many red color water molecules there. The one on the right-hand side, the water in the B side only got 3. So left side got more, isn't it? Higher water potential, that means more water there is A. A will have higher water potential. B will have a lower water potential. So the water will move from A to B. Okay, but not yet finished. Another thing to talk about, or to mention the definition of osmosis is it must go through a selectively permeable membrane. This one, there must be a membrane. If there's no membrane here, it's not osmosis. It must go through a selectively permeable membrane. So therefore, you will see that MUHG, you need to have these 3 sentences or clauses to be able to, to be able to, what do you call that, to give a definition of osmosis. Okay, so finally, you see this is the initial condition before osmosis happens. So you find that your water will diffuse from a region of higher water potential to a region of lower water potential, which is the B side, right? And the final result is you will find the water will move from A to B. So in other words, diffusion, water osmosis is actually diffusion of water. Okay, now you will find that the level of water will not be the same. Okay, it will not be necessarily the same because you have B here, you have sucrose molecule. So it affects the osmotic pressure. So water will go in from A to B. So in the end, you'll find the water level of B will be more, I mean water level more, but the molecules be the same. Look at the number of molecules on the left-hand side. You count the number of molecules, water molecules on the left. You count the number of water molecules on the right, the concentration of water only will be the same. Okay, so this is the result, the final observation that you will see. Water from air will diffuse through the selectively permeable membrane, okay, through B, and this is through osmosis. So osmosis must occur through a selectively permeable membrane. There must be a membrane. Otherwise, it's not osmosis already. And it must be referring to water that is diffusing, not sucrose, not anything else. Okay, so osmosis is only for diffusion of water. It's just like a fancy name for diffusion of water. All right, okay. So next, let's look at this one. This is from the textbook. Yeah, you will see again afterwards. You go and look at textbook. So I've got two solutions separated by a selectively permeable membrane here. Okay, one. This is called a U-tube. Not the YouTube that you watch the channel and all that. You watch my video. U because the shape is a U, right? It's a tube, a glass container that is made into a shape of a U, and then you can put in solution inside. So you separate the two sections of the U-tube with a selectively permeable membrane. You put on the left-hand side a solution that has high water potential. That means more water molecules. So you can count here. The water molecule is the one in blue. So on the left-hand side, you see got how many? All right. Okay, so osmosis refers to the net movement of water molecules. You see on the left-hand side, got 8. Okay, 1, 2, 3, 4, 5, 6, 7, 8 molecules of water. Okay, of course there'll be millions actually, just to show you only. This is just to compare, like let you have an idea. Water in this will be millions and billions, right? Okay, on the right hand side, you see there are only two water molecules here. So you'll see immediately, you'll see which is the high water potential and which solution will have a low water potential. So the 8 one is high water potential, the 2 is low water potential. Okay, so just now you mentioned the concentration of water from more to less, right? Now, so water will move through the selectively permeable membrane. So you will see at the end here, there will be 5 molecules here and 5 molecules here. All right, you see not? All right, and then so this is how water molecules passes through, right? Through the membrane, okay, the plasma membrane, from high water potential to the low water potential. That means which side got more water, that will be high water potential, and the side which got less water will have low water potential. Okay, now there is another definition of osmosis which you can also use. Instead of talking about water, I can also talk about the solute. The solute, that means the substances that are dissolved inside that. So if you look at high water potential, look at this one, right? Just now I mentioned that this solution on the left has got, it is high water potential, but you see it's low solute. Okay, I only have got one, the red one. Let's count the sucrose molecule. 1, 2, 3, 4, 5, 6. I've got 6 sucrose molecule on the left. But you see on the right hand side, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 sucrose molecule. So this is higher solute potential. Okay, so I can also, instead of talking about high water potential to low water potential, I can also modify my definition of osmosis by using the solute word. I can also say, use this definition: the net movement of water molecules from an area, from a region of lower solid potential to a region of higher solid potential. Also can. It has the same meaning. Just now the one we talk about water potential, high to low. This one, we talk about solid potential. If solid potential, that means it must be from low solute to high solute. Because low solute means it's got more water. Okay, less solute means more water. And then more solute means less water. All right, you see the idea or not? If I give you a solution that is very, very, very sweet, a lot of water, a lot of sugar, that means if I compare, sugar is more, then the water is less. So if it's high water, high solute potential, it will be low water potential. And then on the other hand, if I give you a solution that is very, very diluted, okay, let's say manisana, right? I put very little sugar but a lot of water. All right, so this will be high water potential but low solute potential. Okay, you can see the idea there. All right, okay. Now, don't forget your third sentence or third phrase, which is always must say to a selectively permeable membrane. So this is also can be used as the definition for osmosis. Just now I talk about water, okay? This one I talk about water potential. This one I talk about solid potential. Also can. Okay, so here you see, okay, now only water can pass through. Usually it's because of the size. Okay, so you see on the one on the left, this one is higher water potential. These are the smaller ones. You see the small, looks like bees. That one, the blue color thing, that is water molecule. All right, you see more on this side, on the left side. And then on the right hand side, it's got less. Therefore, you see the molecules are moving across from left to right. Okay, and then on lower water potential is on the right hand side. So you can also say on the left hand side, it has got lower solid potential. So now you look at the green molecules here. These are solute. The small ones are water. So lower solid potential here means less of these green molecules. And then on the right hand side, we call this higher solid potential. You have a lot of this green color. I mean the green dots here. That is solute. So water move from, you can say, from higher water potential to a lower water potential, or from a lower solid potential to a higher solid potential. Okay, that means from a dilute solution to a concentrated solution. Okay, that is the idea of osmosis. So water always diffuses from a dilute solution to a more concentrated solution. So in the end, you'll find water will be going through. There's a net movement of water from the dilute solution to the concentrated solution. Okay, clear, right? Now, so you can see here also, osmosis. When will it occur? How long will it take place? It will take place until a dynamic equilibrium is achieved. That means both sides will have equal concentration of water. We're only referring to water. So you see on the left hand side here in the beaker, there's more water compared to the number of water molecules on the right hand side because it has got solid there. So water potential is less on the right. So water will diffuse through until a dynamic equilibrium is achieved. That means water concentration, the difference is 0. So on the left hand side, maybe you have 10. Left hand side 10, right hand side also 10. 10 number of water molecules, that means same concentration of water molecules. Okay, then you call it the dynamic equilibrium is achieved. Okay, but osmosis does not stop. Okay, it does not stop in the sense that the water molecules will doesn't diffuse anymore. Actually, it will. It's just that the rate of moving in and moving out from left to right, right to left is the same. So it looks as if there's no net difference. It looks like there's no net movement. Actually, osmosis still occurs, but the rate of water moving in from A to B is same as the rate of water moving from B to A. That means same, right? When they're same, you don't see the difference in the effect. All right, so there is a net movement of water from left to right here because the left hand side has a higher water potential and the water on the right side here has lower water potential. Okay, clear? Right. Okay, no questions so far. Now let me go on to the next one. The third type of passive transport. So we're still in passive transport. Okay, facilitated diffusion. Now because of the word facilitated, what does it mean? It means aided. That means help. This diffusion is helped by something. Okay, so because some of the molecules are unable to go through the phospholipid bilayer, maybe because it's too big, okay, or maybe it's because it's charged, or maybe because it's polar, so it will go through another place which will help to bring it through. So that is what your transport proteins are for. That means channel protein and carrier protein. So if the substances move across the plasma membrane with the help of these transport protein, either channel protein or carrier protein, this is known as facilitated diffusion. But without energy, yeah? Okay, if it's with energy, it's considered active transport. So it moves across the plasma membrane with this carrier protein or channel protein, but without energy. It's considered facilitated diffusion. Okay, so here it will also same principle as your other types of passive transport. It will also go down along the concentration gradient. That means from more to less. More molecules, you must see which side got more. Okay, the one that has got more will always move to the side that has got less. It will move across through the transport proteins. Does not require energy. That's why it's called a passive transport. And it will continue until a dynamic equilibrium is achieved. It's the same meaning when both sides have the same concentration of molecule. That's what it means by dynamic equilibrium. Equilibrium means it's like balance. Okay, both sides will have same number of the molecules. Just now maybe it's 10 and maybe 20 and 5. And then once it goes through, right, both sides will have equal, let's say, then you have achieved dynamic equilibrium. All right. Now, let's look at things. What are the things that go through this channel protein? Now, channel proteins will form channels or canals. Canal is like a terusan. There's like a space there which allows the things to pass through. This one allows small size solute. So it must be small. If you want to go through the channel protein, it must be small, and it allows ions as well. So water can also pass through. Water and charged ions, they will go through the channel protein because they cannot go through the phospholipid bilayer. Okay, so the channels have specific internal characteristic that allows only specific ion. Not everything can pass through. Okay, only specific ion that is specific for this channel protein can pass through. It's not like general purpose. Not everything also can pass through. It has got specific one. Okay, then next one. The example here is small size. The characteristics is small size. It's either charged or polar, and it's water soluble. So it will go through the channel protein. Okay, the other one, which also uses the transport protein, is this one, carrier protein. They also have specific size. Okay, they only can allow specific substances to pass through. Okay, because they need to bind. Bind means ikat. They need to bind onto a specific area in the molecule, in the transport molecule. Okay, in the transport protein. So example, glucose molecules can only bind to the specific site of a glucose carrier protein. So you don't, like I said, don't have the multi-purpose one. Again, these transport protein are not multi-purpose. They're only specific to carry certain type only. Okay, so what are the things that can pass through carrier protein? Will be this one, large size, charged or polar, and also water soluble. Hello, Devesh. All right, I hope you are doing fine. All right, I eager to get your result coming soon. I think the result will be next month. Judging from past experience, last year the result came in June. I think it will also come next month. All right, must be very, very excited. Yeah. Okay, I hope you will score A+ for your bio. Okay, let me know. Please do message me whatever you get. Okay, just let me know how you did. All right. Okay, I'm sure you'll do fine. Okay, let me carry on. Yeah. Now, facilitate the diffusion through carrier protein. So you need to know how to explain. This is MUHG. MUHG. So you need to. This is one of the things you need to remember. How to explain the facilitated diffusion through the carrier protein. How to explain the entire. This one could be a few marks. Okay, if it's part of the structure, it could be maybe 3 marks or 4 marks for you to explain the entire thing. Okay, let's look at how you explain how the substances can pass the carrier protein. Okay, number 1. So you must see which is more. Extracellular glucose concentration are higher than in the cytoplasm. That means outside. Extracellular means outside the cell. So the glucose is more on the outside. On the inside, the cytoplasm will be less. Okay, so you can imagine it's supposed to come in because outside more, inside less. Okay, so the next thing to talk about, the first thing you say is glucose binds. So the word bind is important. It will bind to a specific site of the carrier protein. So in the carrier protein, there's a specific site for the molecule that you want to carry through to bind there. Okay, so the important word here is bind. You don't see the word, or I don't see the word bind, I don't give you a mark. All right, so the word bind is important. Okay, next. So after it binds there, now look at this important word again. Another important word is it change shape. The carrier protein will be able to change shape. So you can see the shape already changed. Originally like this, when you want to bring it inside, it will actually turn the carrier protein. Like remember we said the phospholipid, this what they call the plasma membrane is dynamic, it is flexible. Okay, the shape of this carrier protein is also can change, right? So it will change the shape and it will allow the glucose molecule to pass through the carrier protein, okay, to enter the cell. So this one also must mention. Binds first to a specific site, and then the entire carrier protein will change shape. Okay, once it change shapes, it will let the thing pass through, right? Then after that, it will return back. It will go back to the original shape. See, this is the original shape, so that they are ready to do another one, to repeat. Okay, the whole cycle repeats itself. So it will carry another molecule and come in again. All right, so these 3 points are necessary when you want to write your answer. Okay, all right, okay, Teresh, remember, let me know, yeah. Okay, I'm sure you'll be happy with your results. I hope you'll be happy with results. Okay, now let's carry on, yeah. Let me carry on. Now we go to active transport now. Okay, so active transport is very similar to your facilitated diffusion because they use the carrier protein, but with a slight difference. The difference here is only in the form of whether it uses energy or not. It uses ATP. For this case, active transport is the same process, right? You just move to the carrier protein, then it will change shape, also the same thing, right? But this one got slight difference. It needs ATP molecule. The ATP molecule will bind onto the carrier protein before this transformation or changing shape can happen. Okay, definition first. Active transport is the movement of, movement of what? All right, movement of molecules, particles, ions, all right, across a plasma membrane, but you must say against a concentration gradient. It is from a region of lower concentration, that means from less. This is less. This is more, all right? By right, it should going from more to less, right? But this one, no. Because the cell requires it. The cell wants this thing. Even though inside already got a lot, they still want to bring in some more. All right, still want to bring in some more. That means if you want to do it, you have to put in energy. Then you will have to pull the thing inside. Okay, so from a region of lower concentration to a region of higher concentration with the use of energy. So, okay, so 3 things here. What is moving? Don't say water. All right, don't say water, say molecules or ions. And then you must say from where to where? Lower concentration to higher concentration. The third point is to mention it needs to use ATP. All right, okay, clear? Now, lower concentration to higher concentration and the use of ATP. Okay, so it is facilitated by carrier protein, must use carrier protein. All right, carrier protein is needed in this to bring it in. So this process will result in the accumulation or elimination of molecules or ions in the cell. So this one will not result in what we call dynamic equilibrium. You will not have both sides equal. Yeah, there will not occur whereby you bring it inside and then both sides the same. No, this one will not, because your cell needs it. So you will have one side will be always getting more and more and more. That's why you call it accumulation. Accumulation means getting more and more and more. And then if the cell doesn't require this, it will push it out. All right, it will remove it. So you will be pushing it out. It's called elimination. So you will be either accumulating it more inside or you will be pushing it outside. So that means it will never have occur a situation whereby both sides equal. It will always be one side more than the other. Okay, so this is the active transport. The difference, okay, afterwards you will see the comparison. Okay, it will not achieve dynamic equilibrium. Okay, so remember to say that. Next. So require specific carrier protein with specific size. So remember just now I said Carrier protein is not multi-purpose. You have specific carrier protein only for certain things, for certain substances. So if the substances want to move in, if it cannot move into here, it must go in through another specific carrier protein. Okay, so it has to bind because not all molecules will have the same shape. It cannot fit into all. Okay, there's no one carrier protein that can fit all the types of molecules. Okay, so here you see lower concentration out there, outside the cell, higher concentration inside the cell, and you see that ATP is needed. Okay, so carrier protein also possesses receptors to bind. So this is the receptor. There's a place here where the ATP is bound here. There is a place for the ATP to bind. It's a molecule. ATP is a molecule that carries energy. So when ATP is released or when ATP is broken down, you are going to get ADP and the inorganic phosphate, which can be used to make back the ATP. Okay, so once your ATP is used, it will break down, it will give you the energy. The energy will be used by the carrier protein to change shape. Okay, so that is why you need the ATP. You need the ATP, the energy from the ATP to change the shape of the carrier protein. Okay, so the carrier protein changes shape when a phosphate group attaches to it. Okay, the phosphate group is from the ATP here. All right, okay, so next one, this is the site to bind the ATP molecule. There's a site there on the carrier protein. Okay, so as a result, the molecules or the ions will move across the membrane. Even though it is a lower concentration to higher concentration, you'll be able to pump it in or to bring it inside because there's energy involved. Okay, next one. I need to tell you more because this one is important. Okay, you must know how to explain the entire process. So active transport results in accumulation or elimination. So there's no one side that will be both the same. There won't be both sides same. There will always be one side that is more, one side is less. Now this type of carrier protein, there's another name called pump. Okay, involved in active transport. The carrier protein that's involved in active transport are called pumps. So in our body, there are a few types of pumps. Okay, pump is like, pump up, bringing in something. For example, in animal cells, the carrier protein that transports sodium ions out and bring in potassium ion, they are called the sodium-potassium pump. All right, okay. So this sodium-potassium pump, the function here is to push out the sodium ions and bring in potassium ion. So it's just because the cell needs it. The cell needs the potassium, and the cell does not want so much sodium. So it will be exchanging. So you can see here the sodium, all right, is the blue color hexagon shape here. And then the potassium here is that purple color oval shape here. So this pump, actually, the same pump, it will push out the sodium. I can see here the first diagram here, it attaches the sodium, right? And then the second diagram here, you will see when the phosphate group attach the ATP attach here, it changes shape. And then it will open up and pushes out the sodium. At the same time, before it closes back and comes back to the original shape, it will bring in the potassium. So now two ions of potassium will be, attached onto this sodium potassium pump. And now the shape will change back. Okay, will go back to original, and the potassium ion is now transported inside. Okay, so you see, it's a sodium potassium pump. This carrier protein will push out the sodium, at the same time bring in potassium. So in the end, you will find more and more potassium inside the cell, and less potassium, and more sodium outside the cell. Okay, more potassium inside and more sodium outside, because it is does not achieve an equilibrium. All right. Okay, so here sodium goes out and potassium comes in. All right. Okay, so let's look at this is from the textbook. You will see I've explained to you. Actually, it's the same thing. All right. 3 sodium ions bind to the carrier protein. Okay, this is how you explain. You want to explain this, how does the sodium potassium pump work? You say, first of all, is to bind. So the keyword here, bind. You must say the word bind. Bind to the specific site. Then after that, ATP attaches to it, right? It will decompose, it will break down. So they break down into ATP and P. So the phosphate group will bind onto the carrier protein. All right, and then this will change shape. The phosphate bond provides energy and changes the shape of the carrier protein, so it like moves the other way, and then the sodium ion is transported out, pushed out. All right. Number 4. But at the same time, you see, they want to bring in potassium ion. So the potassium ion will now bind again with the carrier protein. All right. And then the phosphate group will leave the carrier protein. Right. When that happens, it will change shape. It will go back to the original shape. The loss of the phosphate group will restore the shape. Okay, it will turn back this way. And now your potassium ion is brought into the cell, transported into the cell. Okay, so when an exam comes, right, MUGA should say, if the question asks you for this, you just have to memorize what is in the book. Okay, it's all there. The understanding is important. Understanding is what I'm here for, to teach you, to tell you, to explain to you so you understand. But the remembering part, you have to remember on yourself. I cannot remember for you, right? You have to, once you understand, you will be able to memorize. Okay, and you just write all the points that are needed, like highlight all these things, you will be able to get good marks. Okay, because this is what is needed in the scene. All right, now let's look at the animation here. Okay, now look at it carefully. 3 sodium ions attach, the phosphate attaches and it changes shape, then it pushes out. At the same time, the potassium ion comes in. Okay, look at it here. Potassium, when it opens, the potassium comes in. And then now when the potassium ion detaches, then it will turn back the shape. Okay, so this is what the sodium-potassium pump works. It is in your cell. Okay, specific cells in your body. So your cells will accumulate potassium inside, and it will eliminate sodium from the cell. All right. Okay. So one more minute. Just a while. Just let you have a look. One more round. So again, your potassium, sodium attaches. Your ATP comes and binds. Then it moves out. At the same time, your 2 ions of potassium comes in and it changes back. All right. The potassium, the phosphate is removed or released. Okay. Now, so this is another pump. Another pump is known as a proton pump. The proton pump is on the cell of this, is in the cell of this stomach, the gastric cell, the cells that are lining our epithelial cell lining the stomach. Okay, so this cells actually produces acid. It produces acid, and this is the acid that makes your digestive juices acidic. Okay, these cells will be producing acid into the stomach lining. These cells are found on the epithelial cells, okay, on the stomach, in the stomach there, right? So you see here, the proton pump increases the acidity of the stomach contents. So it pumps the hydrogen ion out. And as you know, hydrogen ion is the one that makes it acidic. So it pumps it out. Okay, the carrier protein pumps it out. So your stomach content here becomes acidic. All right, and you know that it's good because you what they call that, you will kill bacteria in the food. Okay, it kills one of the function of this hydrochloric acid is to kill bacteria. So inside the cell, you have high pH, that means low acidity. You have low concentration. High pH means low acidity. Yeah, low concentration of hydrogen ions. So it will pump out because outside is pushing out your hydrogen ions. So outside here in the stomach there, you have low pH, that means very acidic. You have high concentration of hydrogen ions. Okay, so energy from the ATP enables the hydrogen ion to be transported out by carrier proteins. That means this is the proton pump towards the extracellular fluid, the stomach here. So this is the stomach now. This is your stomach lining. All your juices your stomach juices are here. Okay, and this is the cell that is on the stomach wall. So it pushes out the acid. Okay, so you make the acid inside your stomach wall. This causes an accumulation of the hydrogen ion and acid in the stomach cavity. So the stomach cavity becomes more and more acidic, and that's why you have the hydrochloric acid there. Okay, so summary, right? How to remember? It's important for you to remember the 3 types of transport here. Okay, first of all, if it's lipid soluble, all right, it will go in through simple diffusion. Lipid soluble, the what they call this lipid soluble, the fatty acid, glycerol, vitamin A, D, E, and K, and also steroids. Okay, doesn't matter big or small. Then water can also squeeze through. Okay, water squeezing through here, this will be osmosis. The method by which water passes through is always osmosis. Okay, special name for diffusion of water is osmosis. Okay, next. This is the channel protein, and these are the carrier protein. Now, if you need to use this one, that is considered facilitated diffusion. All right, water-soluble ions will go through the channel protein, and water-soluble and larger molecules will go through the carrier protein. So these two are called facilitated diffusion because no energy involved. Okay, so facilitated diffusion is either channel protein or carrier protein, and it is without the use of energy, but using the transport protein. Okay, and lastly, also this one is called passive. All these three are called passive. Then the other one, water-soluble ions, large molecules, okay, but they want to travel the other way around, from less to more. Okay, this one is going against concentration gradient. And with energy, this one will be known as active transport. Okay, it also uses carrier protein, but it's against the movement, against, that means from less to more. All right, so this is a summary. So remember this, you may want to copy it down later. All right, okay. So lastly, okay, similarities comparing passive transport and active transport. So comparison is also important because you will have a question as you compare and contrast. Okay, compare and contrast. Please do not forget your similarities. You need to have at least 2 similarities. Yeah. Okay, so look, similarities. Both methods, whether passive transport, active transport, both will move substances across the plasma membrane. Okay, you will move things around, either go in or go out. Yeah, that is a similarity. Another one is it occurs across the plasma membrane, which is a selectively permeable membrane. So we'll see the differences afterwards. So this is a similarity. Both active and passive transport will have these 2 common characteristics. Okay, differences. Okay, now differences, passive transport and active transport. That means we're comparing passive transport including your simple diffusion, osmosis, and facilitated diffusion. Okay, so now occurring where? Okay, for passive transport, it can occur in living cells and also non-living cells. That means across your Visking tubing, for example. Okay, Visking tubing allows osmosis. Visking tubing also allows simple diffusion. Okay, it's a non-living cell. But for active transport, it only occurs in living cell. The reason is because it requires energy. And non-living cells will not have energy. So no energy, it cannot carry out the active transport. Okay, next, passive transport does not require energy, whereas active transport will require energy. Okay, in terms of the usage of energy requirement. Next one, what about the direction? Okay, so for passive transport, it occurs following the concentration gradient, or you say down the concentration gradient. That means from a region of higher concentration to a region of lower concentration. For active transport, it occurs against the concentration gradient. That means from a region of lower concentration to a region of higher concentration. You don't have to write both. If you say occurs against the concentration gradient, is enough. You get your mark already. Or you can say occurs from the region of lower concentration to a region of high concentration, also the same. No need to write both. Okay. Lastly, how when is it stop, or the occurring? How does occurs until when? Okay. So for passive transport, it will occur until dynamic equilibrium is achieved. Both sides will have equal concentration of the substance. Okay. But for active transport, it will not achieve dynamic equilibrium. There will always be one side of the membrane which will have more, and the other side of the membrane will have less. That means it will result in either accumulation or elimination. Okay, one side that is less will be means eliminate already lah, means push it out already lah, so it will have less. And then the one side that bring in the thing will be considered as accumulation, getting more and more of the molecules. All right, okay. So this is what you need to talk about. Remember all these points, please. You can write in table form, even in essay, but make sure you write in full sentences. Okay, not just one word. Write in sentences. Easier for the mixer to understand what you're saying. All right, okay. So that is it for the lesson for today. Okay, it's a long lesson, but hopefully I did take the trouble to explain it to you. Now your job is to remember now. Okay, I hope you have understood. But the next thing is you need to remember and you write it out during the exam. All right, okay. MUHG, if you have any questions, you can PM me. All right. Okay, then I'll give you my Telegram. What they call that? Telegram name. Right. You can PM me if you need any you not need to explain anything. All right, okay, students, I will see you next week then. Hopefully, I can get you can get another lesson ready in time, 3.3. All right, so I'll see you hopefully, and thank you very much. Okay, bye bye, everyone. Stay safe and keep hydrated. Okay, bye bye. Thank you, everyone. Have a good evening. Have a good weekend.